Nutritional requirement and nutritional classification. Bacterial nutrition. Bacterial cell has its principal constituent as water, 80%, of total weight. Rest is protein, polysaccharides, lipids, nucleic acids, mucopeptides. Minimum requirement for growth and multiplication of bacteria is water. A source of carbon. Nitrogen. Inorganic salts. Water is the vehicle for the entry of all nutrients into cell and elimination of water. Bacteria require adequate nutrition, optimum pH, temperature and oxygen for the growth and multiplication. Suitable artificial media containing sources of carbon, nitrogen, hydrogen, oxygen, phosphorus and other elements such as sodium, potassium, magnesium and iron in a very small amounts, have been used for the cultivation of microorganisms in the laboratory. Bacteria can be classified depending on nutritional requirements. First, source of energy. Bacteria which derive their energy from sunlight are called phototrophs. For example, Rhodospirillum rubrum. Bacteria which derive their energy from chemical reactions are called chemotrophs. For example, Escherichia coli. Second, source of electrons. All microbes require a source of electrons for their metabolism. Bacteria which use reduced inorganic compounds as electron donors are called lithotrophs. For example, Pseudomonas pseudoflava. Some other species which use organic compounds as electron donors are called organotrophs. For example, E. coli. Third, source of carbon. Microbes requires carbon for synthesizing the cell components. Some species use CO2 as the major source of carbon. These microorganisms are called autotrophs. For example, nitrifying bacteria. Other species requires organic compounds as the source of carbon. Such species are called heterotrophs. For example, E. coli. Fourth, nitrogen. Bacteria can use nitrogen from the atmosphere or from inorganic compounds such as nitrates, nitrites, ammonium salts or organic compounds such as amino acids. Nitrogen is the major component of protein and nucleic acid. Fifth, sulfur. Sulfur is needed for the synthesis of amino acids. For example, cysteine, methionine, etc. Sixth, phosphorus. Phosphorus is an essential component of nucleotides, nucleic acid, phospholipids etc. Seventh, mineral salts. Growth factor or bacterial vitamins and water are also essential for bacterial growth. Bacterial requirements. Oxygen. Carbon dioxide. Temperature. Moist damp, drying. H-ion concentration. Light. Osmotic effect. Mechanical and sonic stress. Oxygen requirement. Division is based on influence of oxygen into growth. Aerobic, require oxygen for growth. Anaerobic, growth in absence of oxygen. Microaerophilic, grow best in low oxygen tension. Facultative anaerobes, ordinarily aerobic but, can grow in absence of oxygen. Aerobic obtain energy through oxidation. Anaerobes use hydrogen than oxygen. CO2 requirement. All require small amount of CO2. Requirement usually met by CO2 in atmosphere. Temperature. Bacteria vary in requirement of temperature for growth. For each species there is a temperature range and growth does not occur above maximum or below minimum. Optimum temperature. Temperature at which growth occurs best at 37 degrees Celsius. Mesophilic, bacteria grow best at 25 to 40 degrees Celsius. Psychrophilic, grow best at temperature below 20 degrees Celsius, for example -7 degrees Celsius. Thermophile, grow best at higher temperature. 55 to 80 degrees Celsius. Extreme thermophilic bacteria can grow at above 80 degrees Celsius. Thermal death point, lowest temperature that kills a bacteria under standard condition in a given time. At low temperature some species die rapidly but most survive well. Moist and drying. Water is essential to bacterial protoplasm and drying is lethal. Treponema is highly sensitive to drying while other withstand drying for months. Spore may survive in dry state for several decades. Hydrogen ion concentration. Bacteria are sensitive to variation in pH. Each bacteria has a pH range, above or below to that bacteria can't survive. Optimum pH are at which bacteria grows best. Bacteria grows best at neutral or slightly alkaline, pH 7.2 to 7.6. Depending on optimum pH value of MO, they can be classified as 1. Acidophiles with optimum pH range between 1 to 6.5. For example, Lactobacillus acidophilus. 2. Neutrophiles. Most bacteria grow in narrow pH range of 6.5 to 7.5. For example, E. coli, Salmonella typhi. 3. Alkalophiles with optimum pH range in between 7.5 to 14. For example, Vibrio cholerae, pH 9.0. Light. Bacteria, except phototropic, grow well in dark. They are sensitive to UV light and radiations. Cultures die if exposed to sunlight. Osmotic effect. Bacteria are more tolerant to osmotic variation than most other cells. Due to strength of their cell wall, sudden exposure to hypertonic solution may cause withdrawal of water and shrinkage of protoplasm called plasmolysis. Sudden transfer from a concentrated solution to a distilled water may cause plasmoptosis, that is, excess osmotic inhibition leading to rupture of cells. Mechanical and sonic stress. Bacteria have tough cell walls. They may be ruptured by mechanical stress such as grinding or vigorous shaking by glass beads. Thank you.